Okay, let's get started. First of all, so we're supposed to prepare some polls, but the way this meeting is configured, I cannot do that. So please ignore the poll icons, and we're gonna use the chat to for you to answer the questions that I'm asking you. So please, it's a very busy chat. So please do not write random stuff. We're gonna go through some housekeeping notes in a second. Okay. So when I wanna discuss with you today, so this is a workshop 16 from UCLA QC Bio Collaboratory. And the workshop is divided into three days. Day one, we're gonna mainly talk about sequencing and the DNA library preparation. While in day two, we're gonna do some specialty DNA library preps and some RNA libraries. And on Thursday, we're gonna experiment a new online lab session. So I'm asking you, can you hear me and can you see the screen just fine? Please answer in the chat. Perfect, great. Okay, so let's get started. So this is the outline of today's class. So we're gonna start with a brief intro on the sequencing technologies that are available today. We're gonna discuss the how to prepare a library, and then some more in details steps of the library preparation. Also, we're gonna discuss in detail, what does sequencing mean and what are the features that you have access to. We're gonna finish today by talking about the targeted sequencing and the PCR duplicates, which I think it's very, very important. And then as you can see on the screen, you see some, let me just use this maybe. You can see on the screen like these type of icons. So these were supposed to be the polls. As I told you before, it's not gonna happen. So I'm just gonna write on the chat the questions. I'm just gonna talk about that, and then I'm gonna write on the chat the questions, and then you can answer. Okay. Also, you can see here we're gonna have some mini quiz, this is just very like 3 questions every time. And then I'm going to take attendance, so it has a dual purpose. And then if we have time, if I think we have time, we're going to perform a Kahoot. If you've never done it, it's quite fun. We're going to have a few questions. This is just the first time that I do it like a completely online workshop. So you're the beta tester today. Okay. So again, I have, if you have some questions, please do not use the chat. Last week I was co-teaching for another workshop, and people were posting their questions on the chat, and then at the same time like 10 questions popped up, and so it was very difficult to basically answer. So what I'm trying to do this time, and let's see if it's working, I set up a Google form, sorry, a Google spreadsheet. I'm gonna share the link with you. And so for each section of the class today, there's a space to ask questions. If the space is running out, please just add more lines, and then you can just write your question here. And then I'm gonna type the answer here, or I can talk about it. I'm gonna still type the answer even after the class if I didn't write it down, because it's much faster if I talk about that. And then if you have more questions, or you're not happy with the answer that I gave you, you can always email me, and my email is actually here in row 3. So you can always email me to ask me more questions, okay? So let me share the link with you right away. Where are you? Here. And so I also want to check whether you can access the link so you can ask questions, okay? So I'm just gonna post the link on the chat, and please let me know if you're able to access it. So I'm gonna give you like a couple of minutes to access the link. And so we can just basically communicate questions through that form, okay? So whenever you are done checking out the link, please just write down, yes, I can. I can see it. If you want to try to type something, sometimes there's problems with the ability to edit. So to just type your questions, that would be great. Okay, perfect. That's what I suspected. Okay, let me modify this. Thank you so much. Interesting. All right, please let me know if now you are able to type in your questions. You can just type in something, you know, just to see if you can edit. I see a lot of, awesome, perfect. All right, thank you so much. So we can just get started. So again, no questions in the chat. It's going to be too crowded, so please try to use the Google Sheet. I have it open, so I can answer that. You can use the chat just to report some tech problems like audio, video, if for whatever reason I'm disconnected and stuff like that. And please make sure your mic is muted if it's not already, okay? Again, post your questions in Google Drive, and again, I will answer them. Post during the class, unfortunately, we're not going to be able to do that. So forget about this. We still have to figure out some way of doing this more efficiently. And this class again will be recorded, and the videos will be available soon. Okay. All right. So let's get started with some intro to sequencing technologies. Okay. I just want to pop up this figure because I just want to tell you that everything is interconnected in NGS. So you may have taken some classes about methods for analysis, for example, RNA-seq or ChIP-seq or bisulfite sequencing or variant calling and stuff like that. But everything is interconnected. So my workshop is the only one that it's more like kind of experimental. And as you can see, the type of library preparation, which is the blue box, is affecting how you do the analysis, but also the sequencing is affecting how you prepare your library, and the type of starting material is affecting how you do your library, right? So everything is totally interconnected. So that's why I think we decided to include my workshop in the offer of the QC bio, just because the analysis part is not sufficient by itself to perform a NGS experiment. Okay. So in this very dense slide, there are like the three main categories of sequencing. So the first generation sequencing you most likely know about is the Sanger sequencing. So the Sanger sequencing approach is so-called the first generation sequencing. Also, the Maxam-Gilbert type of sequencing is considered to be the first generation sequencing. In general, long reads, a lot of material, it will basically give you like the consensus sequence of the mixture of DNA that you have um, in the um, um, in the sequencing reaction. Okay, so the second generation sequencing, which was very important, especially in the scaling up of the sequencing of the human genome, it's the next generation sequencing, which is mainly like the two leaders in the market are Illumina and IonTorrent. Okay, actually, about like 90% of the sequencing is performed using Illumina. So that's what we're going to talk about today, um, and we're going to discuss how how the sequencing is performed. Also, I just want to mention some few features of the next generation sequencing or second generation sequencing technology. An amplification step is required before performing the sequencing reaction itself that occurs into a sequencing machine. The technology behind it, it's called sequencing by synthesis. We're gonna see that because basically you are adding a nucleotide at each sequencing cycle, and then you are basically imaging the nucleotide that has been incorporated, okay? So that's why it's called sequencing by synthesis. Usually the reads are short. We're talking about below 500 base pairs total, but it can produce millions of reads, okay? So we're talking about, 100 millions, even billions with the new sequencing machines of reads per run, okay? So it's a very, very high throughput, okay? The error rate is very low, and however, since it require amplification before sequencing, it cannot read native modifications, okay? In contrast, the third generation sequencing technology that you see highlighted in red in the slide has different features, okay? First of all, it has a high error rate. However, new library preparation techniques basically reduce this error rate to something that is very similar to the Illumina, if not better. Okay, so this is it's true, the sequencing itself has a higher error rate. However, there are ways of getting rid of it. The number of reads, it's much lower. We're talking about thousands and low millions. It can read native modifications, okay? The reason why it can read native modifications is that it does not require amplification before sequencing, okay? You can perform amplification before sequencing. You can also skip that. So in that case, it will give you native modifications, which is really useful, especially if you are looking at modifications at DNA or RNA. So there are very few methods that can do RNA modification detection this way. The reads are usually longer. We're talking about kilobases and also megabases have been reported. So one single molecule can be read as 1 megabase or even longer. And again, I told you, you can sequence single molecule, okay? So the signal that it's giving you, it's coming from one single molecule and it's real time, okay? So as soon as the read is read, you have data available, okay? And the two main players of these third generation sequencing are PacBio or Pacific Biosciences and Oxford Nanopore, okay? So the two technologies are totally different. I'm not gonna go too much in detail about this, but the Oxford Nanopore, it's basically a portable instrument. We have it in the lab. It's it's very cheap and you can use. It's very easy to use. However, PacBio requires like a huge, gigantic machine that is very, very expensive. Okay. However, they have advantages and disadvantages, but they have more or less the same features that I mentioned before. Okay. All right. So let's go back to the Illumina technology, which is the technologies that most of us were gonna end up using and the leader on the market right now. So how the Illumina technology works is basically you have your input DNA, and what we're gonna see today is this part right here. So how to prepare a library for sequencing, okay? So I'm not gonna talk too much about this right now, but you can basically see that you are adding pink and purple pieces of DNA next to your DNA fragments. And then you basically load the mixture of these modified DNA fragments into the sequencing machine. And what you can see is that this is basically part of the sequencing machine, this glass slide. And on the glass slide, you have these primers, let's call it primers or oligos. They are complementary actually to the pink and purple sequences that we attached during the library preparation. And the presence of these primers allows you to perform the so-called bridge amplification, okay? So this is a method to basically amplify your DNA, okay? So that's what I was referring to before. You have to perform amplification before sequencing in the Illumina type of library preparation. And the reason why we do have to amplify is that the technology is basically imaging in each pieces of these, in each piece of this glass slide, the signal, okay? So if the signal is coming only from one molecule, the detector will not be able to image that signal. However, if we perform an amplification, this bridge amplification, we're able to create a much bigger signal from that particular spot. And so at that point, the detector is able to get the signal from these molecules, okay? As you can see here, it's called bridge because it forms bridges during the amplification. And so in this step, you see you have one molecule that ends up being two molecules right here. And then if you repeat this process many, many times, you end up having the so-called colonies of library, okay? So you basically have from one single insert here, you have many, many fragments at this step. And so what happens after you amplify your fragments in this bridge amplification, you see that all the fragments are actually next to each other. So that's the beauty of the system. So you basically create amplicons that are basically next to each other, okay? What happens is you're adding one nucleotide at a time, A, T, C, or G, and these nucleotides are fluorescent, okay? And they're also blocked. So you can only add one at a time. Once you're adding one nucleotide, then the machine is basically scanning, okay, of what nucleotides you have. And then once you scan the entire glass slide, then you can determine what type of nucleotide has been added to that particular spot, okay? And then you repeat this cycle many, many times. And as you can see at the bottom right of the slide, so this is, for example, let's say cycle 1, and this is cycle 2, this is cycle 3, 4, and so on, okay? So if we look at, for example, this spot right here, okay? So it starts with a G, and then there's a C, T, G, and A, okay? So this is giving us the sequence of this particular fragment, okay? And that's how it works, okay? All right. Any question on the Illumina sequencing technology? All right. So in this slide, I just want to show you and introduce you to some nomenclature that we're going to talk about later today. So these are top left. You have depicted the some of the instruments that the Illumina has. The most widely used was actually the HiSeq. Now everything is being sequenced, or most of it is being sequenced on the NovaSeq, and there's like some smaller instruments that are still used, the NextSeq and the MySeq. But for example, if we take the NovaSeq, the HiSeq as an example, you see here at the bottom left, this is called the so-called flow cell, okay? So the flow cell, it's basically the glass slide that I was talking about before. Is actually slightly different, but you can see that this glass slide is divided into 8 parts, okay? So 1, 2, 3, 4, 5, 6, 7, 8. and each part of the flow cell, it's called a lane, okay? So the minimum part of the Illumina sequencing machine, it's called a lane. So that's a minimum, yeah, I would say part. And then if you sum up some lanes, then you end up with a flow cell. Okay, so this is the nomenclature of flow cells and lanes. Okay, usually when you sequence one library, you just sequence one lane. So, but you know, depending on the application that you are performing, you might sequence more than one lane and even an entire flow cell for some applications. What I was telling you before about the glass lights and the primers that were just, you know, a lot of primers on the glass, it's actually not true anymore, because Illumina has introduced these pattern flow cells. So if you imagine that these are basically just extremely small wells, and in each well there are like these lot of primers, and the amplification occurs within each well. Okay. Now the advantage of this format is that the machine doesn't have to scan and trying to understand, okay, so here I have a group of fragments, I want to get the signal out of it. Now this is every microwell is ordered in a specific coordinate, so the machine knows exactly where to look at. Okay, so it's much faster. So this was introduced in the high-seq and the Nova-seq type of sequencing machines. Okay, so the advantage is that this allows you to sequence in a much faster fashion. The drawback of this, we're gonna see it later today. We're gonna have problems using this format because of index swapping. Okay, but we're gonna talk about this specific problem later on. Okay. So let's, okay, so if you have questions about the Illumina sequencing, I'm just gonna check. Okay, so I see that someone has a question. Okay, let's wait for the question. So whenever you are done, whenever you're done writing your question, please move your cursor away from your question. Otherwise, I'm not going to be able to see it. Okay. Each lane as each sample. Okay. So we're going to talk about this actually later on. You can have one sample per lane. You can actually have multiple samples per lane. You can have one sample over multiple lanes. Okay, so this depends a lot on the amount of sequencing depth that you need for each sample. Okay, but we're going to discuss this multiplexing issue later on. So the answer is yes, but also, you know, you can have different configurations. Okay. How the machine knows each layer of nucleotide? Does it wash the first block, then continue? How the machine knows each layer of nucleotide? Does it wash the first block, then continue, or how it happens? So, so it's basically it's adding, you know, it's adding nucleotides at the same time, and then basically each nucleotide is called the reversible termination fluorescent nucleotide. So it basically, the polymerase can only add one nucleotide, and then after the addition of the polymerase, then it washes everything out, and then the machine is scanning for the presence of fluorescence. Okay, and then after the machine finishes the scan, then these reversible terminator nucleotides are basically deprotected. So now the polymerase can add a new set of just actually one nucleotide, and then the cycle basically goes on and on and on. Okay. All right. Let's see if there is one extra question. So whenever I finish answer your question, if you can type in the chat, I don't know, you know, thanks, this answer my question, or, you know, something like that, so I know that I don't have to talk about that question anymore. Thank you so much. how many molecules can be in a flow cell? Okay, so this is a good question. How many molecules can be in a flow cell? A lot. So each flow cell actually can be different. Um, we are probably going to talk about how many molecules, how many reads a flow cell can produce later on. But again, it ranges between, hundreds of millions to basically billions of molecules, okay? If the question was how many molecules do you insert in a flow cell, that is, it depends on the size. Uh, usually the sequencing core will take care of it. But if your question was how many molecules can you get out of a flow cell sequencing, then yes, it's between hundreds of millions to billions per flow cell, okay? All right, okay. Looks like we are good for this. So let's go ahead. Let's go ahead with the presentation. Okay, so let's get started with the library preparation. Okay, so how do we prepare a library? Okay, oh, one more thing. If I don't see your question, or if you want me to answer my question, please raise your hand. Um, um, if you click on participants on the bottom of your Zoom, I think you can easily raise your hand, and then I will try to answer. If I'm not seeing you, just write something in the chat. Okay, don't write the question in the chat, or just say, Marco, you have a new questions, and then we can just go ahead. Okay, so let's get started. Library preparation. How do you prepare the library? Okay, so what I usually like to when I want to talk about how the library preparation works is I usually compare it to a um, um, bike ride. Okay, so I actually used to live in West Hollywood. So if you imagine, and I am the starting material. So for example, a gDNA, and what I want to have is the final library that it's at UCLA. Okay, so during the bike ride, we're gonna have different steps. And if you look at the top left part of the screen, these are the main features that are needed for a library to be sequenced on the Illumina machine. Okay, so it has to be DNA, 100% every time. This is a second generation sequencing, so it has to be DNA. It has to be short, so it's usually between 200 to 800 base pairs in terms of size, and it has to be modified with the adapters. Okay, so if you remember from the previous slide, you saw these two pink and purple pieces of DNA. These are called the adapters. Okay, we're going to talk extensively about that. Okay, so how do we do that? Okay, so we have our DNA, and as you know, the genomic DNA that you extract from cells is like, it's huge. It's megabases. It's the smallest that you can think of. It's the DNA of viruses, which is still kilobases. So for example, the coronavirus, the new coronavirus that it's creating such a problem right now, has a 30 kb genome. Okay, so even if it's RNA in that case. Okay, but in general, the viruses are the organisms with a smaller DNA, and it's still kilobases. So we are still like one order of magnitude above the limit of the short DNA. Okay, so what we usually do is we perform fragmentation. Okay, so we fragment the DNA and we make it smaller. Okay, in about that range. Okay, so we already have DNA, we fragmented. Okay, so part 1 and part 2 are already satisfied. Okay, and then we just have to add the adapters. Okay, and the step that we use in the lab to add the adapters is called adapter ligation. Okay, so it's using a DNA ligase enzyme, and at that point we have our final library. Okay, however, the fragmented DNA, it's usually not compatible with the adapter ligation, and this is the same as having a cop stopping you because you didn't follow the street rules. Okay, so what we actually have to do is we add an additional step. It's called N-repair and A-tailing. Okay, so this is basically a step that makes the fragmented DNA compatible with the adapter ligation step, and then at that point we can have our 3 features basically satisfied. Okay, I've added also the concentration and QC at the beginning and concentration and QC at the very end of the lab preparation. Okay, so we usually perform at least a concentration measurement at the beginning of our library preparation, and then at the end to just check if the library worked okay. All right. So what I wanted to ask you right now is if you can write in the chat. Again, this would have been a poll question, so we'll be able to see the percentage. But what is the method that you use for DNA quantification? Okay, so as of now, we are preparing a library. So the simplest library preparation that we're performing is a DNA library preparation. Okay, so let's pretend we extracted the genomic DNA from our cell of interest. What type of method to quantify your DNA do you use? Do you use nanodrop? Do you use agarose gels? Do you use, I don't know, qubit? Can you just quickly type it in the chat so I have a sense of what you guys are doing? Okay. All right. So I see a lot of nanodrop and qubit. That's great. Okay. So it's great. We are gonna see in the next slide which way is the best one. Okay. So most of the times the answer is nanodrop, and the nanodrop is actually a spectrophotometer. Okay. It's actually pretty bad to quantify the DNA. Okay. So if I'm telling you, do not use the nanodrop to quantify your genomic DNA. It's only good, and I still use it, by the way, to quantify or basically to check the quality of my extracted DNA. Okay, and it's good because it's a spectrophotometer, right? So we can check basically the 260-80 ratio and the 260-30 ratio. Okay, and as you can see, the 260-80 ratio is telling us whether we have a contamination from proteins. Okay, in that case, if we have it, then we can basically perform a protein digestion, for example, to just get rid of it, or a phenylchloroform extraction. Also, the 260-80 ratio is telling us if we have a contamination of RNA in our extracted DNA. Okay, so usually, if it's the perfect DNA, it's between 1.8 and 1.9 of 260-80 ratio. If the ratio it's above 2, so it's like 2, 2.1, 2.2, this is basically a suggestion that you have a contamination of RNA in your preparation. Okay, so in that case, if you want to extract DNA but you have RNA contamination, you can always treat it with RNAs to degrade the RNA and have a pure DNA sample. Okay. Also, the 260-30 ratio is telling us if you have contamination of organics. So, for example, phenylchloroform or other carbohydrates. Okay, and this is only true. So the pure DNA, it's usually above 2. If it's below 2, it means that you have contaminants. Okay, so I usually use the nanodrop just to check the quality for the presence of contaminants. And again, it's okay to use the nanodrop if you want to measure the RNA. Okay, so for RNA is quite accurate, but for DNA measurement is not very accurate. Okay, so I saw some of you wrote down nanodrop and qubit at the same time. So if you're using the nanodrop and qubit the way that I just described, it means that you're doing a very good thing. Okay, the qubit is the best way to measure your DNA. Okay, the advantages of using the qubit is that it's more accurate and specific. It's a fluorometer. Okay, so it basically, we're going to see this in the lab in day 3. We're going to add a dye that it's specific to our DNA, our RNA, our proteins, if we want to measure proteins. So it's a dye specific to this type of molecule, and that's why it's very accurate, because it's just measuring that molecule for you. Okay, and it's also good for low concentrations. Okay, so sometimes the nanodrop, if the concentration is below a certain threshold. I usually prefer, for example, not below 50 nanograms per microliter. I don't use the nanodrop. However, for the same reasons, for the same advantage that the qubit has, the problem with this is that it does not detect contaminants, okay? So it's very specific to your DNA molecule or RNA molecule. However, is not telling you that you have contaminants. So that's why I usually perform the measurements with both the qubit and the nanodrop at the same time, okay? Using both is giving me both informations, okay? Again, you can always use a very old method, the agarose gel, to check for the to quantify your samples. And in this case, it can tell you if you have RNA contamination. So for example, if this is a genomic DNA that it's been extracted, the red arrow is pointing to a smear of a smaller size, and this is usually RNA contamination, okay? Also, there's another QC step that you can do, this is done using either the agarose gel or the TapeStation, and this is seen at the bottom right, okay? And this is telling you, not only the concentration of the DNA that you have, but also the quality of your DNA. Is it high molecular weight, like you see, in the far right lanes 1, or it's very degraded, like you see at the far left, like in lane 15, okay? So for some application, it's really important to have high molecular weight. So for example, the third generation sequencing approaches that I was talking about before, PacBio and Nanopore, they usually are able to read longer molecules. And so for some applications, you wanna have very high molecular weight. So you may wanna have a preparation of genomic DNA that it's more similar to lanes 1, 2 versus lanes 15 or 14 that are very degraded, okay? So this is another type of quality control that you can perform depending on the type of preparation. For the preparation that we're gonna talk about today, the size of your DNA is not really important because the first step, and we're gonna see it right now, it's fragmentation, okay? So even if you have a high molecular weight DNA, the first step is just to make it smaller, shorter, and so you don't care too much if it's high molecular weight to begin with, okay? All right, so as I told you, we have our purified DNA, we quantified, we checked whether we have basically contaminants, so we remove all the contaminants, hopefully, and so we are able to just perform fragmentation, okay? So there are like 4 main types of fragmentation that you see here in the slide. Nebulization, I'm not gonna talk about it. It's nobody uses it anymore. We're gonna focus our description in step 2 and 3, sonication and enzymatic, in the next slide. I just wanna mention the chemical fragmentation. It's used mainly for RNA fragmentation, so we're gonna talk about this tomorrow. So I just wanna mention that, but that's another type of fragmentation. But the 2 main ones are sonication and enzymatic, okay? The main difference is sonication, you need a machine. So for example, you might have heard of Covaris, you might have heard of Bioraptor. So these are the 2 main sonication systems available on the market, but there are many others. And this is a physical type of fragmentation. So it basically just fragment the DNA independently of the sequence. On the opposite side, you have the enzymatic fragmentation method. And enzymes, they recognize the DNA. There are very few enzymes actually in nature that are aspecific. So all the enzymes have somehow a specificity of substrate. And most of the times, the enzymes that deal with DNA or RNA, they have a sequence specificity, okay? So like one very easy example that I can make of enzymatic fragmentation is the restriction enzymes. So for example, EcoR1 or HIN3, these are all enzymes that are able to fragment and cut the DNA, okay? However, these enzymes, they have a very specific sequence that they recognize, okay? So it's not very good for NGS applications to have this sort of very biased method of fragmentation. So what the companies have come up with is somehow engineer and produce a mixture of enzymes that are, less specific than restriction enzymes. And so they call it, like, for example, fragmentase enzyme mix, okay? And they basically can fragment the DNA in somehow an aspecific way, okay? So there's still a little bit of bias. As you can see on the plots A and B on the right, you see, so the lines are basically the percentage of each nucleotide, TCGA. And as you can see, the physical type of fragmentation, you have, basically no bias whatsoever, while in the enzymatic type of fragmentation, even if it's using these engineered or, basically studied mixtures, you still have in the first few bases, we're talking about the first 5 or 10 bases, you still have some bias for each position, okay? So it's not extremely unbiased, but it's very close to being unbiased. Why people want to use enzymatic methods for fragmentation? Well, it's an enzymatic, it's basically an enzymatic reaction. So you can just incubate your DNA in a PCR thermocycler, and then you can perform many at the same time. While for sonication, since you need to use an instrument, most of the times the instrument has slots for 1 to 10, and sometimes very expensive instruments have also 96 possible wells for fragmentation, but they're very expensive. Okay, so it's a very cheap way of basically using the enzymatic method to fragment the DNA, especially if you want to have high throughput type of library preparation. Okay. However, again, you will suffer for these minor biases at the beginning of the read. Okay. All right. So I told you before. So we have DNA. We've started with DNA, so we're good. Our DNA is short. Okay. So we've used either sonication or enzymatic fragmentation for that, and then we have to modify the DNA with adapters. But I told you before, the fragmented DNA is not compatible with the adapter ligation, okay? We have to perform this intermediate step that it's called end repair and A-tailing. However, in order to understand why we're doing these steps of end repair and A-tailing, I just want to talk about a little bit about the adapter ligation and how the adapters are made and how to make the fragmented DNA compatible to the adapters, okay? So we fully understand why we have to perform this intermediate step, okay? So these are the Illumina adapters depicted here, okay? Why are they important? Okay, so they carry a lot of information on them, okay? So these are basically two oligos, two DNA oligos. They have a lot of information, okay? So let's take a look at the information that it is encoded in Illumina adapters, okay? So the first important information that they carry is the sample multiplexing. So, okay, so this goes back to the same concept that I briefly mentioned during my answer to the question about how many libraries can I sequence in one lane, okay? The sample multiplexing is actually allowing you to tag each DNA with a different barcode, okay? And the barcodes are basically just short sequences, okay? So just, usually between 6 to 8 to 10 nucleotides of DNA that are basically present here, index 2 and index 1 right here, okay? So for example, for sample 1, one of the two barcodes or indices, you can use the term interchangeably, it's ATGCGC, and then you have the other index with a different sequence. And sample 2 will have a different sequence for each of the two indices, okay? So as you can see here, whenever you have 3 samples, one sample will be tagged with the purple indices, one sample will be tagged with the blue indices, and the other sample will be tagged with the orange indices, okay? So it's a way of basically using the combination of both index 1 and index 2 to differentiate the origin of each DNA. Okay, and the information is encoded here in the green box, and it's index 1 and index 2. All right. The other information that is encoded in the Illumina adapters is the red box right here. You see it as called a P5 and P7. This is just the nomenclature of the Illumina adapters. And these are called the amplification regions, okay? So they're really important because on P5 and P7, we usually have our PCR primers. So at the end of lab preparation, most of the times we perform a PCR to increase the amount of material that we have. And our PCR primers are designed exactly on the P5 and P7 regions, okay? The other important, and I would say the most important thing about these P5 and P7 regions is that they are necessary for flow cell binding, okay? If you remember, at the very beginning, we had the purple and pink DNA molecule at the very ends that were complementary to the primers of the flow cell, and then you can see it actually at the bottom of the slide right here. In this case, they are actually colored as orange and green. So these are exactly the regions that the flow cell has complementary oligos designed to, okay? So if you don't have these orange and green regions, then your fragment will not be able to bind the flow cell. It will not be able to form these colonies of molecules. You will not have enough signal to be detected by the Illumina machine, okay? So basically these are really, really important for the sequencing step, okay? So we have our sample multiplexing, we have our amplification regions, and then we have, let's take a look at the shape of our adapters. So if you notice, they're not fully double-stranded DNA. If you tilt your head on your left 90 degrees, you see a Y, so they're called Y-shaped, okay? And the reason why they're Y-shaped is because now they carry more information, okay? So this region is complementary. This has one index, okay? The dark brown has one index, the red has the other index, so you have the ability to have two indices encoded. If they were fully double-stranded on this part, you could only have one index. And then you can have both P5 and P7 information on the same side. But why didn't we do like a, you know, instead of like using one Y-shaped partially double-stranded oligo, why didn't we do like just two fully double-stranded oligo, okay? And the reason is depicted here, okay? So let's pretend on the left we have two oligos, and each oligos, you know, the orange one has all the P5 and the barcodes that I described before, and the P7, the green one, has all the P7 and the barcode that I described before. So in the case of the left, you have linear double-stranded DNA, and in the case of on the right, you have the Y-shaped Illumina adapters. Okay, so on the left, you see that whenever you're doing ligation, so whenever you're attaching these oligos to your fragment, which is depicted in black here, you have these possible combinations on the left with linear double-stranded DNA. You have orange, green. You have green, 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 orange, and orange, orange, okay? But the correct information, it's only when you have both included, okay? So only in 50% of the cases, you have the information of both P5 and P7. While if you look on the right, you have a Y-shaped type of adapter. Every single time, you can try to draw these schematic as many times as you want. Every single time you will have 100% of the fragments will carry both the P5 and P7 information, okay? So this is just a way of getting the libraries with the correct information in a much more efficient way, okay? So that's why we usually don't do linear double-stranded DNA adapters, but we usually use Y-shaped adapters, okay? So the final very important information that is carried on the Illumina adapters, and it's actually the one that we are mostly interested in this step of the library preparation, is the end of these adapters. Okay, so this is depicted in the blue box. Okay, you basically see that you have a T3' overhang. Okay, so on the opposite side there's no nucleotide, and the 5' on the opposite side is phosphorylated. Okay, so this is exactly how it looks like. Okay, so you have your adapter, and then you have a 3' T overhang in this part, and then you have a 5' phosphorylated on the other side. I'm asking you a question, so this would have been a poll question. How do you think you will need to modify your fragmented DNA in order to be compatible with these Illumina adapters? Okay, so you have a T overhang. You need something on the other side that is complementary to the T overhang, and also remember you have a phosphate group here at the 5' end. Okay, if you have somehow a molecular biology background and you know how the DNA ligases work, you should be able to answer this question. You can try to guess it on the chat if you want. I'm just gonna give you like 1 minute to think about it, and then I'm just gonna explain it to everyone. Okay. All right, looks like nobody wants to attempt the answer. Restriction enzymes. So this is all right. So Karina said 8-tail oligohands. That's a good start. It's actually not sufficient, but it's a very good start. So your fragments need to be 8-tailed. Okay, so in this case, you basically have some complementary, something that is complementary to the T overhang. So that's really good. However, there is one more thing that it's really actually really important, and without it, you will not be able to perform ligation. And that is actually you need a 5' phosphorylation. Okay, so your insert needs to have a 3' A overhang. These will basically match the 3' T overhang. However, in order to complete the ligation, you need the phosphorylation on the 5' end. Okay, so you have the 5' end phosphorylation on the adapter already. You need the 5' phosphorylation on your insert as well in order to bind these two fragments. Okay, so these are the two conditions that we need in order to make our fragmented insert compatible with the ligation part of our Illumina adapters. Okay, it's possible to use restriction enzymes to make it compatible, but then if we use restriction enzymes, then they need to have, you know, somehow to be unbiased, you know, and they need to produce a 5' phosphorylation, which is usually what they do, and a 3' A overhang, which is not, you know, very common restriction enzyme. But you could theoretically use restriction enzymes for this purpose. Okay, but again, the main approach that it's used here is 5' phosphorylation, A overhang, and to our insert, and that makes everything compatible. All right. So that's exactly what we're gonna do. It's called the steps are called end repair and A-tailing. Okay. This is a very busy slide. I just wanna introduce the enzymes that we're using in this step sequentially. So if you start from the left here of the slide, you have two different type of fragments. Okay, so in the top case, you have a 5' overhang, and at the bottom you have a 3' overhang fragment. Okay, the first enzyme that we're using, it's called T4 DNA polymerase. Okay, and the activity of this enzyme is blunting. Okay, so it can fill in 5' overhang, so that's why here you see that everything is basically blunt. So in this case, we are adding a nucleotide, which is a T. And at the bottom, you know that the DNA polymerases are not able to extend in a 3' to 5' direction. Okay, so how can you basically extend this part to just make it compatible with the overhang G that you have here. Well, in this case, the T4 DNA polymerase is actually acting as an exonuclease. So it's chewing up these G and C to just make it blunt. Okay, so in this case, you have blunt end DNA, not by addition like it's performed in the top strand, but just by exonuclease function. Okay, the result of this enzyme reaction produces, doesn't matter how it was at the beginning, it produces blunt ended DNA. Okay. After that, another enzyme comes into play. It's called T4 polynucleotide kinase, or PNK for friends, and it's basically just phosphorylating the 5' ends and dephosphorylating the 3' ends. Okay, so the activity of T4 DNA polymerase and T4 PNK is actually blunting and phosphorylating and dephosphorylating in order to make it compatible with our adapters. Okay. The one thing that is still missing is the A overhang, and this process is performed with the A-tailing enzyme, and this can be a DNA POL1, derivative, which is called clenoexo minus, or a Taq polymerase. So I don't know if you know that, but whenever you're doing a PCR amplification with your Taq polymerase, your final products will have a overhang at the very ends. Okay, so we can incubate our blunt and phosphorylated fragments with a Taq polymerase, and then these will add preferentially A's at the end, and they are gonna be overhangs. Okay, so this is exactly what we were looking for. Okay, so it's a blunt with A overhang, 5' phosphorylated fragment. Okay, and this is compatible with our adapters. Okay, so now we just mix everything together, and here you are. You have your adapters, Illumina adapters. Oops, sorry, my bad. You have your Illumina adapters here on both the left and the right, the Y-shaped Illumina adapters, and then you have your insert. Okay, and now everything is ready for the T4 DNA ligase. That's the enzyme that we're using to attach the adapters. Okay, to basically seal everything together. Okay, now we have our final library. Okay, so do we have DNA? Yes, we already had DNA to begin with, so that was always checked out. Is it short? Yes. We perform fragmentation. Is it modified with adapters? Yes, it's modified with adapters. We just ligated our adapters onto it. Okay, and with this, I'm gonna stop for 5-10 minutes. So we're gonna come back at let's come back at 2:50. So we have time to stretch and drink some water. And in the meantime, if you have more questions about the first part or the second part, I'm gonna answer them right now. Okay, so I'm just gonna go and take a look at the Google Drive questions right now, and then we are coming back at 2:50 sharp. Okay, so 2:50, I'm gonna start again. Okay, so the question is, if I already tried, if I already finished the DNA preparation, could I try RNAs at the end? Yes, you can do that. I mean, there's no disadvantage in doing that. There's actually no advantage in doing that. Usually, you want to remove the the RNA before starting the library preparation, because sometimes the RNA is interfering with the library preparation steps, and so your best approach would be remove the RNA before starting the library preparation, and then start the library preparation, with a clean template. But you can still remove the RNA at the very end. I just don't see the point in doing it at the very end versus doing it before starting the library preparation. All right. I hope everyone is back. I just want to check if we are recording. Yes. Okay. So let's go ahead and add more details to the library preparation steps. So what we've talked about up to now, it's how we did fragmentation and repair a tailing, adapter ligation, and most of the times actually we are doing a PCR at the end of the library preparation. Why we do that? Well, the first, the easy answer is we need more material for sequencing. Okay, so sometimes we have very limited amount of original DNA, and so we just need to perform a PCR to have more material. However, the PCR is not creating you new molecules. It's not giving you more information. It's just making the information possible. So what is the PCR doing? Is just introducing biases. So it's just copying existing molecules. Okay, so it's not creating new information. It's just like duplicating the information that you already have. And so with this, it's introducing biases. Okay, so not every fragment will be amplified at the same rate during the PCR. And with this, it's creating duplicated reads. Okay. Again, I just want to stress this out. Are the duplicated reads useful? Yes and no. Okay. Yes, because it makes your library to be enough concentrated to be loaded on the Illumina machine. However, it's not creating new information. So that's why I call them useless. Okay, so it's not giving you information. It's just making the information that you have compatible with the Illumina sequencer. Okay. So if you have to do the PCR, then go ahead and do it. But the fewer PCR cycles, the better. Okay. There's also the possibility to monitor with the qPCR what is just the right amount of PCR cycles that you need to have enough material for the Illumina sequencing machine, but not too many to create too many duplicated reads and a lot of PCR biases. Okay. There's also another reason sometimes that you need to perform a PCR. So what we've talked about up to now, it's a strategy where we have the full-length Y-shaped adapters for Illumina sequencing. And the full-length Y-shaped adapters means that you have both P5, P7, the two indices, index 1, index 2, on each side, okay? But sometimes some type of library preparation, they have these so-called stubby Y adapters, okay? So the stubby Y adapters are basically adapters that they do not carry the entire information, okay? So they don't have the indices and they don't have the P5 and P7 information, okay? So we can still introduce both the indices and the P5, P7 information during our final PCR. We just design a primer with the 5' overhangs having this information and a complementary sequence to the stubby adapters for amplification, okay? So this is a second reason why sometimes we have to do the PCR, not only to increase the amount of material. Sometimes you have to do PCR because you want to introduce the indices and because you need more material, okay? So it's a double reason. All right, so as we discussed, we have our final libraries. Once we are done with our libraries, we perform a QC, okay? And this QC basically, it's usually performed by checking the size distribution, presence of adapter dimers and primer dimers, and we're gonna see it in a moment. And these, and also in the lab, this can be done using a Agilent TapeStation or a regular agarose gel. However, we're gonna see why the agarose gel is usually not a good method for checking the size distribution. Also, we're gonna perform concentration measurements, and we're gonna use the qubit in the lab. But another very popular and very accurate method for final library concentration measurement is actually the qPCR, okay? So qPCR, we're not gonna see it in the lab just because it's gonna take too long. but it's another method to check the final concentration of our final libraries. It's actually superior to the qubit because it's only measuring the molecules that are amplifiable. Okay, so it's superior to the qubit in this sense. Once we have both the concentration and the size distribution, we can dilute our library after calculating the molarity, which is a measure of concentration. And then we are ready to submit our samples for sequencing, okay? So let's basically tackle one point at a time. So we have our final libraries, okay? So it's we have different, you see like there are like different type of libraries, okay? We have A, B, and C, okay? So each pair, or three in case of A, they have more or less the same distributions. So I'm just asking you, and please answer in the chat, which one do you think it's the best library, A, B, or C? You might know it, you might not know it, you know, just try to guess. Just type it in the chat whether you are able to see it. Which one do you think it's better, A, B, or C? All right, it's probably written in the next slide. So you guys cheated. I'm pretty sure you cheated because usually I have some A's and B's among the answers. But yes, so basically the best type of library preparation is C. Okay, and I'm gonna explain you the reason why. Okay, so both PCR primer dimers that you see in A and adapter dimers that you see in B are considered to be contaminants. Okay, of your final libraries. Your final libraries can, you know, you see the final libraries up here. So these are usually smears, okay? So it's a smear, it's not like a definite band. It's a smear of, you know, fragments of different sizes, okay? However, the PCR primer dimers are much shorter and the adapter dimers are also much shorter than your libraries. If you see your final libraries are going between 200 to, you know, 800, 1000. While the adapter dimers, erase here, while your adapter dimers are usually between 120 to 150 base pairs, and the PCR primer dimers are usually much shorter than that. We're talking about 50, 60, 80 base pairs, okay? And why am I saying that these are contaminants, okay? Usually we aim to have less than 1% of both adapter dimers and primer dimers, okay? The reason it's very simple. First of all, you are not wasting reads, okay? So if you look at the adapter, I mean, if you imagine the adapter dimers are just basically two adapters ligated next to each other, and they have all the features to be able to be amplified, okay? They're short, they're double-stranded DNA, and they have the P5 and P7 that are needed for Illumina sequencing, okay? So they can be sequenced. It's just that the sequence does not carry any information. We already know the sequences of the adapters. There's no insert within the adapters, so we don't have any information. So if I'm reading a lot of adapter sequences, which I already know the sequence of, I'm just wasting money, okay? Wasting reads and wasting money, okay? So this is the first reason, okay? And the second important reason why I don't want to have these type of contaminants is because if you have adapter dimers or free adapters or free primers in your final library, so if you didn't perform a good purification at the end of your final library, you are gonna have index misassignment or index hopping problems. What that is basically, you know that the adapters or the primers or both carry the information of the index or barcode, and the index or barcode is telling you, okay, this fragment is coming from my sample 1 or sample 2 or sample 3 or sample whatever, okay? So if I have index misassignment, and this is just like basically a byproduct of the amplification that occurs in the Illumina sequencing, one index which is derived from free primers or free adapters in your final library, can basically be attached to a fragment coming from a different sample. So, for example, I have DNA from sample 1, which is supposed to have index 1. However, I see it as index 2 if I have index hopping. And so I'm saying, okay, so I see this fragment which has index 2, it must be coming from sample 2. It's actually not coming from sample 2, it was originally from sample 1. So if you have this index misassignment, you can basically say, okay, this fragment was coming from a different sample that it was supposed to be, okay? So if you minimize the presence of adapter dimers and primer dimers, this problematic occurs less frequently, and so you will have much cleaner results at the end of the day, okay? So these are the two reasons why we need to have less than 1%. I just wanna stress this out. That's why we are using, advanced electrophoresis system like TapeStation or the bioanalyzer you may be familiar with, instead of agarose gel. Because if you think of that, we need to be able to detect 1% of our adapter dimers and primer dimers. If you run an agarose gel, the ethidium bromide resolution, it will basically show a band only if it's at least 10 nanograms. Okay, if it's 10 nanograms and it has to be 1%, it means that our original DNA that we have to load in the agarose gel in order to be able to see 1% as adapter dimers, it's 10 times 100. Okay, so it's 1 microgram, which is a huge amount. We are not going to have micrograms of DNA at the end of the library preparation unless we do a lot of PCR cycles, which is bad because we are introducing a lot of biases. Okay, so that's usually why we prefer to use these machines, because it allows us, they're very sensitive, so it allows us to basically load a very limited amount of input material, and so we don't have to do too many PCR cycles and introduce biases. Okay, so that's why usually you can do agarose gel electrophoresis, but we usually don't do that. Okay. I just briefly mentioned, if you didn't purify your final library well. So, and this brings me to how do we purify our libraries, or how do we purify our reactions during the library preparation? So we talked about, end repair, A-tailing, adapter ligation. How do we purify our DNA after each reaction? Okay, there are two main methods. One is, the spin column. It's very similar to what you usually use for extraction of genomic DNA from your cells or your tissue. So we basically mix DNA binding buffer with our DNA. We load it on a spin column, okay? This one, which has basically a filter, let's call it like filter, that is able to bind to DNA in the presence of the DNA binding buffer. Then we discard the eluate, okay? So, we discard the flow through, which is basically, usually contaminants, proteins, and buffers and stuff like that. We wash it just to remove, further remove contaminants, and we add, an aqueous solution, and now the DNA detaches from this column or filter, as you want to see it, and then now we have purified DNA. However, there's a drawback of using the spin columns. So spin columns are actually very fast. Usually, like these type of purifications, you do it in 5 minutes or so. There's a drawback of using the columns. They're usually losing quite a lot of material during this step, and also they retain fragments that are very, very short. For example, they are able to retain double-stranded DNA down to 20 base pairs. Okay, if you remember, our contaminants, they're actually much bigger than that. So if I have a column that retains everything that it's above 20 base pairs and up, yes, I retain my final library, but I'm also retaining adapter dimers and primer dimers. So I'm not able to purify them out, okay? So that's why in the lab, we usually use the so-called AMPure beads or spry beads. They have different names, different commercial names, but they're basically magnetic beads that are specifically coated with a polymer that is able to bind DNA in the presence of these PEG and sodium chloride buffer, okay? The concept is very similar to the column. It's just like in this case, everything is performed in solution, okay? And we're gonna see this in the lab, the purification. I think it's one of the most important steps of the lab preparation. So we basically mix the beads and the binding buffer, it's called a binding buffer, with our DNA. Then the binding occurs. And now instead of doing centrifugations to separate the two, we use a magnet, and the magnet is basically pulling the magnetic beads that have our DNA bound to it. We perform washes, okay, just to remove the contaminants, and now we add an aqueous solution, and at that point, the DNA will detach from the beads, and now we have our pure DNA. So why do we use AMPure beads, which usually takes longer? So if the columns takes about like 5 minutes, AMPure beads takes usually between like 30 to 45 minutes for the purification. There are like two reasons. One, the AMPure beads are much more efficient than column purification, so they retain a bigger amount of DNA, and also they have a cutoff. Okay, so the ratios of the buffer that we usually use, and they are highlighted here as 1.2x, 0.8x. They usually they're not able to purify fragments efficiently that are below 150 base pairs, okay? And if you remember, our contaminants are actually 150 base pairs or smaller, okay? So whenever we are performing a purification using, we usually use a 1-to-1 ratio, which means, one volume of beads, for example, 50 microliters of beads and 50 microliters of our DNA solution that we want to purify. It's not able to purify our contaminant size, okay, which is actually here, okay? So using the AMPure beads allows us to do two things. One is removing the contaminants, and two is basically purifying the reaction and getting the DNA pure ready for the subsequent reaction, okay? And so that's why in the lab we prefer to use the AMPure beads, and that's what we're gonna see in the lab on Thursday, okay? When do we perform purification? The workflow that we've seen before, it's considered to be kind of like old, but I chose it because it was easier to follow, to understand. There were like 3 steps of purification, which made the protocol quite long to perform, about like 4.5, 5 hours. However, what we're going to do Thursday in the lab, it's a streamlined workflow. And so it performs fragmentation with an enzymatic enzyme, end repair, A-tailing, and adapter ligation without purifying our sample. And so we basically skipped one purification step here, and we only have one purification after the adapter ligation. We perform a PCR, and then we're gonna have another purification step at the very end, okay? So these are usually the steps where we perform a purifications in our DNA-seq libraries, okay? All right, I'm gonna quickly check if there are any questions in the Google spreadsheet, and then we can continue. Continue from life 15, is there a possibility to affect the quality? Okay, so we have some questions. Let me share the screen so we can all look at the questions. All right, so I'm gonna so Young, I'm gonna answer your question later. Okay, we'll come back to that later. I'm just gonna answer some questions about the library preparation 102 steps. So from line 27 on. What is the difference between lane 1 and 3-4 in C? Slide 36. Why only lane 1 has lower size band? All right, 36. Okay, so this was the slide of the final libraries. And what is the difference? The only difference is that there were like three different type of library preparation. Okay, and basically um, um, basically different samples and three different type of library preparation. Okay. Why only lane one has lower size band? Okay, lane one, or all the libraries that are tagged with A. So if you guys go to, let me do it actually. If you guys look at the lane one or A, why do they only have PCR primer dimers? So this type of library preparation was performed using a kit that used column purification. Okay, so the column purification was able to retain very small PCR primer dimers. So that's why you see PCR primer dimers here. B was performed with a regular kit using AMPure bits. However, the amount of adapter dimers was so high that also the AMPure bits were not able to remove it in just one purification. So we usually, when we see this, we perform an additional purification to get rid of the adapter dimers. Samples C, they were performed beautifully, so no presence of adapter dimers or primer dimers in this region, and so we don't need to perform additional purifications. Okay. All right. Is there a difference? Some just questions from Catherine. Is there a difference between different brands of beads? Yes. So the formulations are different. In general, the behavior is more or less the same. Some of the beads are more expensive. Sometimes they behave better than other beads, but it's might not always be true. You can also make the beads in your lab because they're quite expensive, but the features in general are quite similar. So you can test them on the latter DNA that you have. For example, for agarose gel, that's a great testing DNA. But usually for these type of purifications, they're usually quite okay, all of them. Okay. Why is RNA contamination a problem? What step does it interfere with when it get purified away during preparation? Yes, it might. I can give you an example of when the RNA it's a problem. For some protocols that we're probably not going to be able to see it here because it's it's too much detail. The first step, it's an enzymatic digestion using the restriction enzymes of our DNA. And sometimes if you have a lot of RNA in the prep, that can interfere with the proper fragmentation of the restriction enzyme. Most of the times it doesn't really matter. Of course, we are performing reactions on nucleic acid. So if you have if the majority of your preparation was supposed to be DNA, but it's actually RNA, it's still a nucleic acid. It's not the same molecule. It's different, yes, but it can create problems with the enzymes if you have if the vast majority of your DNA, of your starting material, it's coming from RNA. It's usually not a big issue having RNA, but again, for some application it might be. Okay. Is there anything we can do? I'm answering the question from Hifang. Is there anything we can do during the library preparation to minimize primer dimer and adapter dimer contamination in the final library? Yes, absolutely. First of all, the ratios of the adapter dimer to your DNA. So if you're starting with a very limited amount of DNA in your library preparation, if you use a ton of adapters, you might have more adapter dimers. Same for the PCR dimers. Okay, so if you use a ton of PCR primers during your final amplification, and you don't have too much material to begin your PCR with, then you might have PCR primer dimers. If you don't know how much material you have, or if you're not sure, you can always mitigate that with good purifications during the AMPure beads purification. Okay, so usually the AMPure beads purification will take care of the problem, unless for some specific type of libraries that are usually problematic, and usually the low input libraries are the ones that suffer more for these adapter dimers and primer dimer contamination. We're actually going to see on Thursday. So on Thursday, I'm going to prepare two libraries. One, it's following the regular protocol, and the other one is pushed to have more adapter dimers in the final product. And one of the two ways of pushing in having more adapter dimers is actually using 4 times more adapters during the adapter ligation step. Okay, so I'm pushing to have adapter dimers by increasing the amount of adapters. But we're going to see that on Thursday. Okay, so let's go back. Let's go back here. Okay, so I'm gonna share in the chat a link. Please, it's just like three very stupid questions about the library preparation. So I have a sense whether I explained, some concepts correctly or not. This is not graded, okay? But if your final quiz is going to be held on Thursday, will have some problems, I can just go back and just take a look at your mini quiz to see if you did well, and then I can add some points using these mini quiz questions and answers. Okay, so I'm just gonna share the mini quiz. Also, it's very important that you do the mini quiz because I'm taking attendance. So if you don't do it, I will not be able to take attendance for you, which means it's really, really bad because you will not be given attendance, especially if you're taking this for credit. Okay, so if you're taking this for credit, it's really, really fundamental that you do these mini quizzes. All right, and then so it's 3:17 p.m. We are going to start at 3:25. Okay, so 3:25. It's a very, very small quiz. Okay, so I believe you can do it. And then if you want more time, just write in the chat and we can extend it. Okay, so we're starting at 3:25 p.m. Okay. And if you have problems accessing the Google Forms, please write in the chat. Okay, so I'm seeing the first I'm seeing the first coming in. They're pretty good. All right, 3 more minutes, and then we'll start again. We still have about an hour left. All right, let's start. So now we're going to talk about sequencing. Okay, so we prepared the library in the lab. So now what do we do? Okay, so we want to submit for sequencing, but to a sequencing core. But then the core is asking us, okay, so how do you want us to sequence your samples? And then you say, I don't know. And then that's why I'm trying to teach you what each term of the sequencing mean. Okay, so if you look at the top, um, you see this is our final library. Okay, so you have, let's start from the left. You have the P5 region. Okay, then you have the index 2 region, which is also called I5, and for whatever reason the index 2 and I5 is next to P5. Okay, so that's just jargon. Okay, it doesn't really matter. And then you have read one. Okay, then you have basically your fragment. Okay, and so whenever you're doing sequencing, you are sequencing from this particular end that starts from read one. And then you continue. So after your fragment, you have the index 1, which is also called I7, and then you have the P7. So it's a complete fragment. Okay. On the opposite side, you also see read 2. Okay. So whenever you want to sequence single end, you're only sequencing read 1. Okay. So you're basically sequencing from the first base of the fragment that it's next to the P5 adapter. When you want to sequence paired end, you sequence both read 1 and read 2, which means that the same fragment is sequenced from both ends. Okay, so this is concerning the single end and paired end type of sequencing. Okay, so do you want to sequence only your fragment only on one end, or you need both end information for sequencing? Okay, on top of that, the sequencing core is asking you, okay, so how did you multiplex the sample? Do you want to have single index or dual index type of sequencing? Okay, So this depends on how many indices you have in your libraries, okay? Sometimes your adapters, they are basically single indexed, so they will only have index 1, but they will not have index 2, okay? Sometimes your adapters will have both index 1 and index 2, okay? So if your adapters only have index 1, then you're gonna sequence single index, okay? If your adapters have two indices, so index 1 and index 2, or I7 and I5, they are gonna be sequenced dual index, okay? Because you need the information from both indices in order to distinguish the sample, okay? So I just wanna make it clear right here. The two different sequencing modes are independent to each other, okay? So the sequencing of the insert is telling you, is it sequencing single end or paired end, okay? And in this case, read 1 or read 1 plus read 2. And then the sample multiplexing type of sequencing, it's sequencing the barcodes, okay? So these are two different areas of your final fragment, okay? And then the sample multiplexing can be done as single index or as dual index, okay? So just wanna make it clear. You can have any possible combination of the two. You can have single and single index. You can have single and dual index. You can have paired and single index. You can have paired and dual index, okay? I just wanna make it clear, these two are two unrelated things that you have to tell the core in order to sequence your library, okay? Okay, Marco, thank you. I understand the naming, but when should I use single and 50 or single and 100? So here I have a little table. It's indicative. That's what usually people do, but as long as you have a DNA library, you can sequence it, the way you want, okay? So usually people perform single and 50 when they have a ChIP-seq library, RNA-seq library, and small RNA-seq library, okay? Usually people that have a small RNA-seq library, they only sequence it as single and 50, while RNA-seq, you see it here, you also see it here, okay? It really depends on what you wanna study, okay? You wanna study RNA-seq because you have, just study the transcriptome, or you wanna do something more specific, you wanna study the splicing of the transcriptome, or you wanna assemble a new transcriptome from scratch, okay? So in this case, you have different type of sequencing modalities, okay? For DNA methylation studies, we usually sequence single and 100 because you'll see it tomorrow. We're gonna modify our DNA and we need longer fragments in order to have a better mapability. But this is just a very specific case, okay? When do I use paired end 50? So I need to have the information from the second end as well on top of the first end. Usually for ATAC-seq, which is a very common application now, you need the information of both the read 1 and read 2. So that's why we do paired end. High-C, which is basically another type of library preparation which measures the contact between different parts of the genome in a 3D space, requires paired end sequencing. Sometimes, actually, ChIP-seq for specific application can be sequenced as paired end. Okay, so ChIP-seq can be sequenced also as paired end 50. Okay, when do you want to use paired end 100 or longer? Usually when you do specific RNA-seq application or whole genome sequencing or whole exome sequencing. Okay, and we're gonna talk about why we are doing this later. But the short answer is paired end 100 is cheaper per base. Okay, so single end 50 is gonna be the cheapest sequencing. Okay, we're using fewer reagents. Okay, so you can easily understand that. We're only basically using reagents to sequence 50 bases here. Paired end 100 uses more reagents. Okay, so I'm doing 100 bases here, and I'm doing 100 bases here, okay? However, it's cheaper per base, okay? So, for example, if single N50 sequencing costs me $1,000, okay? The paired N100, which has 4 times more bases, because you have 100 plus 100, so it's 200, it's not 4 times more expensive. It's probably 2 times more expensive, okay? So the cost per base is actually half. But we're going to discuss that later with a very simple example, okay? The same table is actually done in a different way. So now I'm focusing on the application that you have. So whole genome sequencing or DNA library. You can sequencing any length. Again, the longer the better, just because it's cheaper per base, okay? So that's why I usually recommend to start from 100 per den, okay? ChIP-seq, it's usually sequenced single N50, but again, I told you, it can also be sequenced paired N50 for some specific application. And also, you see for each application, how many reads do you actually need, okay? So in this case, you need about like 20 million reads or 30 million reads, okay? And this is actually really important to understand, to know how many reads you need, because that is telling you what is your strategy for sequencing multiple samples in one single lane, okay? But we're gonna come back to the multiplexing later, okay? RNA-seq, again, you can have single N50, and for example, for differential expression of transcripts, you only need 20 to 25 million map reads. Or you can have, you know, if you're interested in splicing de novo transcriptome, then you need many, many more, okay? So depending on the application that you wanna do, you can have different type of reads requirements, okay? ATAC-seq, 50 million reads, usually per den 50 or more. High-seq, you need actually a lot of reads for this specific application. We're actually not going to discuss it today. It's a very specific application, but you know, if you need it, you have at least, you know, an idea of how many reads you need. Methylation studies, depending on the protocol that you use, you need, you know, few reads, 10 millions to 20 millions for a specific DNA methylation protocol. Or if you do the DNA methylation in the entire genome, then you need quite a lot, basically, okay? The other question that the sequencing core is asking you sometimes, if they're a good sequencing core, they're asking you, okay, what type of sequencing machine you want to use, okay? So this is a much more difficult problem to answer. Usually, like I would say, if it's a new experiment and if it's a regular type of library preparation, so nothing, you know, very special, go with the latest instrument. It's usually cheaper, better, faster, and you know, if you have to perform other experiments in the future, at least you know that you're using the latest machine, and most likely it will still be around because Illumina is changing the machines quite often, okay? So for example, the HiSeq 4000 is still around, but the reagent cost is much higher than it was supposed to be. So if you're starting a new experiment right now and you sequence on the HiSeq 4000, now you're paying more for the reagents, and maybe in 2-3 years it will be discontinued and nobody will have it. And so you will have data coming from 2 different instruments, and you know, they're not very comparable to each other, okay? Not only you will have biological batch effects, you'll also have technical batch effects because you're using 2 different sequencing technologies. I mean, the sequencing technology is the same, but they're like minor changes in the chemistry, which, you know, it's a big problem, okay? So that's what I usually recommend you to go to. Each instrument has you know, a specific chemistry. That was I was mentioning that. They're usually okay. For some application, for example, for the bisulfite sequencing, you might have some problems using some type of chemistries, but you know, for regular DNA sequencing or RNA sequencing, most of them are okay. Illumina develops these instruments especially for DNA sequencing and RNA sequencing, so you're usually okay for regular applications. All right. All right, so let's go to this very important step of the sample multiplexing. So sample multiplexing is basically the strategy to combine multiple libraries, which means multiple DNA derived from different samples in just one single run, or now you know, in one single lane, okay? Why are you doing this? Okay, so why do we wanna do this? I just wanna point out with a very stupid example, that the higher the throughput of the lane that you're using or the machine that you're using, the lower the cost per base that you're paying. Okay. So the example that I usually give it is like, so you want to buy some pizzas for your friends. Okay. You have a party over. You used to have a party over. Now you don't have parties anymore because of the COVID, right? So you go to the pizzeria and then they're asking you, okay, what type of pizza you want? Okay, so you wanna order, let's say you have like 5 friends, you wanna order like 5 small pizzas, you wanna order basically 3 medium, you wanna order 2 large, or 1 huge pizza, okay? Let's say everybody agreed on the ingredients. So you wanna have exactly the same pizza. Everybody wants to have exactly the same pizza. If you look at the cost, the absolute cost for the pizza, the cheapest is, oh, where are you going? Sorry. The cheapest is actually the small one, okay? Oh, where is my mouse going? Okay, sorry. The cheapest one is the small one, okay, absolute. However, if you compare the relative cost per square inch of your pizza, the cheapest one is the large one, okay? So the idea is, since I'm sequencing, I just wanna have reads out of my sequencing machine, I'm going with the cheaper, okay? Here's another slide that basically has an example. This is an example using one of the sequencing machines that Illumina has. And you see basically that the output of the flow cell here at the bottom is increasing, okay? So these are 3 different type of flow cells that the machine can run. The output increases and the cost per human genome decreases a lot, even if the cheapest is still the smaller one and the more expensive in absolute cost is the S4 or the latest one, okay? However, the cost per human genome, it basically is half with the largest sequencing flow cell, okay? If you have only 4 samples, then it's better, of course, to go with the cheapest one, okay? But if you have, let's say, 48 samples, it's much easier to go with the huge one, which is like cheaper per genome to sequence, instead of like repeating multiple times the small one, okay? That is much cheaper, and you will save basically half of the cost in reagents if you go with the big one. All right. And how you do that? You basically mix samples with a different barcode or indices. Okay, so this is called sample multiplexing. Okay, so if your samples have different tags, and they have tags during the you're adding tags during the adapter ligation or during the PCR. We've seen it before. Now you can basically add many samples in the same sequencing lane, okay? And this will give you the ability to just pay less for the same results, okay? I hope you understood this sample multiplexing and why we are usually trying to multiplex more samples in one lane. And now we are stopping for 5 minutes to just answer questions, okay? So I'm just gonna switch back to the Google form and answer some questions, okay, about the sequencing. Okay, see a question coming in. Can we have no index sequencing? Yes, you can. However, you are only limited to one sample per lane. Okay, you cannot have multiple samples in one lane if you don't have any indices. Um, Nowadays, the adapters that sometimes the adapters comes with the kit, or sometimes the adapters you basically design your own. They're usually designed with indices. Most of the times there are single index, so they just have one index. Sometimes they have dual indices. But yeah, you can have no index sequence. However, I just want to point this out because it's a very particular example. But if you want to prepare a library with no indices, you have to be careful. Sometimes the sequencing cores, they are adding to your sample another DNA library that has no index, and this is just added to increase um, the complexity of your library. It's called the phi-X control library, and that library does not have any index. So if you have a library with no index, and for whatever reason the sequencing core is adding the phi-X in your library, which still has no index, then you will not be able to distinguish the fragments that are coming from your sample compared to the fragments that are coming from this control library that the sequencing core has added. The phi-X is a bacteriophage genome, so it's definitely not human or not mammalian, but there might be like some sequences that you can misassign and mismap between the two um, libraries if you don't have any indices. So what I usually suggest is I would always go with indices, and on top of that, you will see tomorrow when we are gonna talk about the best practices um, in NGS that I actually recommend to use unique dual indices. But that's argument for tomorrow. Okay, so we're gonna discuss this more in detail tomorrow. All right. Awesome. I don't see anybody that wants to type in other questions, so I'm just gonna move ahead. Oh, and sorry, I have to share the screen. Where are you? Here. There you go. Okay, so I just wanna tell you that the library preparation that we just described is not the only way to do DNA sequencing. You also have this this type of preparation, which is actually very popular. Um, it's called next zero library preparation, or tagmentation, if you want to call it. It uses an enzyme. It's actually a transposon from a phage that is actually able to fragment the DNA and attach a DNA sequence at the same time. Okay, so if you remember, our library preparation is fragmentation and repair phosphorylation, A-tailing, adapter ligation, and PCR. This type of library preparation starts from purified DNA, and then it's just one enzymatic step, and it fragments and it adds the adapters at the same time. Okay, so it's a very fast, especially if you have a lot of samples. This will save your life because it's just one enzymatic reaction. As we discussed before, it's an enzyme. So this transposase has been engineered to have fewer biases, but it still has some biases. Okay, so if you remember one of the first few slides that I showed you, where we were comparing the sonication and enzymatic restriction digestion, you still have the bias coming from the enzymatic fragmentation, but it's very minor. Okay, so especially if you have thousands of samples, these might save your life. After this enzymatic step that it's called tagmentation, so it's fragmentation and adding the tags or barcodes at the same time, adapters at the same time, you have to perform the amplification. So this is actually necessary. You have to do it. With the previous step, if you had enough material at the very beginning, you didn't have to perform the PCR at the end, just because, you know, at the end of the adapter ligation, you had everything that you needed for sequencing, P5 indices and P7. However, in this case, you have to perform the amplification just because how this transposon is acting on the naked DNA. Okay, so you have to perform amplification, but you know, it's only 2 steps, you know, you can actually go directly from tagmentation to PCR without doing an intermediate purification. So it's literally like 2 steps, and the final purification at the end, compared to, you know, fragmentation, it's 1, and repair, phosphorylation, and A-tailing, now they're combined, and also adapter ligation, and then you have purification here, PCR, and then purification at the end. So it's much faster. Okay. All right. So again, fragmentation and adapter ligation at the same time. All right. Since it's quite early, we can try to do Kahoot. So if you go to kahoot.it, we are gonna try to do like a fun quiz. Okay. This is not recorded. This is not giving you grade. It's just like, you know, a fun way to review some concepts. Okay. So go to kahoot.it. Just give me 1 second. There you go. Okay. All right. So I'm just gonna share. Where are you? Here. Okay. So go to kahoot.it. Sorry. In your browser, cell phone, or computer, whatever works better for you. And this is the game pin. So you can just, as soon as you're prompt with the, you know, just type in this pin code, and then you will be automatically added to the questionnaire. Okay. I'm just gonna wait like one more minute to have everybody logged in. So the how Kahoot works is, you know, there's a question. Okay, on your device you will see only 4 colors. Okay, so red, green, yellow, and blue, and you have to click on the color that you think is the correct answer. Okay, and the score that the program is giving you is not only based on the if you got it right or wrong, but it also the timing. Okay, so the faster will get a higher score than the one that answers correctly with a with a longer time. Okay, so let's give it like 1 more minute, and then we can get started. This is the first time that I do it completely online. Usually it's quite fun to do it like in the class, but let's see how it works. All right, seems like you know, most of you guys logged in, so let's get started. Okay, so have to get it right first, and then but then you have to be as fast as possible. All right, so let's get started. All right, are you ready? Okay. First question, which step is not required after sonication before ligation? Okay. Which step is not required after sonication, before ligation? Is end repair required after sonication, before ligation, PCR, in vitro transcription, A-tailing? Remember, not required, okay? Okay. And then I think this is also fun because, you know, we have a real-time answers, and then we can review the concepts. So if all of you made mistake, we can just go back and review the concept. Awesome. Okay, so most of you got it right. Okay, so end repair and A-tailing are required after sonication, before ligation, but the question was tricky. So the question was not required. So both PCR and in vitro transcription are not required between sonication and adapter ligation. Okay. All right, let's go to next one. All right, Rockstar Rhino leads, and Fuzzy Puffin is very close to it. Okay, so let's get doing it right. What information do you use to separate DNA sequencing libraries when sequenced in one lane? Okay, so we just discussed it. Okay, what information do you use to separate DNA sequencing libraries? UMI, sequencing read 1, sequencing read 2, indices or barcodes. Hopefully you get it right. This is very important, okay? Okay, all right, awesome. So most of you got it right. So the correct answer was indices or barcodes, okay? Let's address sequencing read 2. Sequencing read 2 is reading your fragment, and then your fragment is not telling you from what sample was coming from unless you're sequencing multiple organisms in one lane, which is, you know, unusual. So usually the indices or barcode is the one. UMI, we haven't dealt with them yet. They actually can separate libraries, but they're usually single cell RNA sequencing libraries, or, you know, single cell DNA sequencing libraries. So it's a more specialized application that we haven't dealt with. But usually the UMI that we're using in regular DNA sequencing libraries will not tell you from which sample they were coming from. Okay. Right. Ooh, Fuzzy Puffin is up top. If I'm sequencing paired and 50, okay, so paired and 50, you have overlapping reads if the insert is how many base pair long. So overlapping means that read 1 and read 2 are overlapping, okay? 50, 99, 101, 151, okay? And there are two correct answers. Okay, so both 50 and 99 are correct. So if you imagine read 1, it's long 50, read 2 is long 50, okay? If my dark gray fragment is long 50, do I have overlap? Yes, read 1 and read 2 will be fully overlapped. If the gray fragment is long 99 nucleotides, do I have overlap? Yes, by 1 nucleotide, okay? So it's 50, and then my read 2 will basically start here, and so I have overlap because the sum read 1 and read 2 is 100, okay? If it's 101, do I have overlapping? No. So I will have like 1 nucleotide that is not covered, okay? If I have 151, it's the same. I have 51 nucleotides that are not covered, and so they're not overlapping, okay? Okay, so Friendly Dog is up top. Next one is probably the last one. Yes. What is the usual size of contaminants in a standard DNA library preparation? Okay. Remember the contaminants that we've talked about. Okay. We have primer dimers, adapter dimers. Okay. So these are the two main. So there are two correct answers. Okay. And then the other two answers are actually the size of library preparation, okay? I hope you get it right. This is really important. All right, most of you got it right. So 50 base pairs, around 50 base pairs, these are usually primer dimers. They're not very common in library preparations. So they're like adapter dimers, which are usually around like 130 base pairs, are more common as a form of contaminant. 250 is not the correct answer because basically 250 is usually a size of our final library. Okay, so let's see who won. First one, third, it's Focused Oryx. Second, Fuzzy Puffin, and the first one is Friendly Dog. All right, good job. And all right, unfortunately, you didn't win anything, but keep trying next time. We'll have another one tomorrow, okay? So now we're gonna switch gear and talk about some different methods for preparing DNA libraries, okay? Today we're gonna focus on target sequencing capture, which is usually performed whenever you want to sequence mutations. And a good example, it's exome-seq. I don't know if you're familiar with it. So you're only sequencing basically the DNA that it's coming from coding regions in your genome, and all the other specialty sequencing approaches we're going to see tomorrow because we're not gonna have time. Okay. The target sequencing capture, I think it's very useful because I want to introduce the UMI concept, the one that you saw in the Kahoot before. And it will give us the ability to talk about PCR duplicates a little bit more in detail, okay? So how does the target capture sequencing work, okay? So the idea is I don't want to sequence the entire genome, okay? Because I'm interested only in, a very limited region of my genome, okay? So you don't want to spend too much money on sequencing something that you don't want to sequence. So what we usually do is we prepare a regular DNA library preparation, like the one that we talked about before, and then we perform this step. It's called hybridization capture, okay? So we have probes, okay? So you have to synthesize, or a company will do it for you. Probes that basically are complementary to the regions that you want to sequence, okay? And usually these probes they can be RNA or they can be DNA, and they are modified to have a biotin molecule attached to it, okay? So they're biotin-related RNA, or again, it can be DNA probes that are complementary to the sequence that we want to have results want to sequence, okay? So we basically mix our library with our probes, okay? And we perform this step. It's called hybridization. So we usually incubate the mixture for a few hours at a certain temperature, usually like around 60 degrees, 65 degrees Celsius. And during that step, you will have the binding of the probes to the target sequence of your interest. And what happens next is you add streptavidin beads to this mixture, and the streptavidin has a high affinity for your biotin, okay? So you're gonna basically create a complex that is streptavidin magnetic beads that are bound to biotin, which is covalently bound to your probes, that is, annealed to your sequence of interest, okay? So just by using this complex, you are able to purify only the fragments that are of your interest, okay? However, you see, you're basically doing a selection, okay? So let's say you're starting with, a huge genome, and then you want to sequence only a very small fraction of it, you're focusing your sequencing efforts to it, which is good. However, you're also reducing the amount of DNA that you have at the end of your library preparation, okay? So you're discarding like a lot of material that you prepared during your library prep, okay? So, for example, whole exome sequencing is only capturing 1 to 2% of your entire genome, okay? So if you imagine your final library was 100 nanograms, okay? Now, if you're capturing, let's say you're capturing efficiently with 100% efficiency your targets, you only have 1 to 2 nanograms, okay? Which are usually, which might not be sufficient for sequencing, okay? So what do you usually do? And this is a question for everyone if they want to answer in the chat. What do you usually do to increase the amount of material that you have of your final library? What operation do you want to perform to increase the amount? Okay. All right. Sounds good. Exactly. PCR. So you basically do PCR amplification, and so this will basically increase the amount of final library. Okay. And that's exactly what the next slide suggested. However, as you remember from before, whenever you're doing PCR, you're basically copying the same molecule over and over and over just to have enough for sequencing, and so you're creating PCR duplicates. Okay, so after you basically sequence, how do you distinguish a PCR duplicate from a true biological molecule? Okay, so that's when the UMI comes into play, okay? So there's actually two ways of getting rid of the PCR duplicates, or to tag the PCR duplicates, okay? So one is UMI independent, and you see it here on the left, and this is just basically an in silico, so like a computational approach. And this is much better whenever you're doing paired end sequencing, because you have the information of both ends. So on this example right here, this is a very rudimentary genome browser example. So on the bottom, you have basically the position of in the genome. Okay, so it's chromosome 1, and this is the position. So 100,000 to 103,000, whatever. And these colored bars are actually the reads. Okay, so reads, read, 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 and read. Okay, so how do you remove the PCR duplicate using a computational approach? Okay, well, you can say, well, if they start and end at the same position, they might, be PCR duplicates. Okay, because that's exactly what the PCR is doing, is copying one fragment from the beginning to an end, exactly as it were. Okay, so you see the fragment red, yellow, and dark blue. These they start and finish at the same position. Okay, so you think, okay, so these might be PCR duplicates. Okay, so I'm just, instead of counting these as 3 separate ones, I'm just saying, okay, I have one. Okay, I have one purple, I have one between red, yellow, and dark blue, I have one green, and then I have one light blue. Okay, so the total number of molecules for this region is actually 4. Okay, however, this might not be true. Okay, so that's how the UMI was introduced in the library preparation. So first, a definition of UMI. So it's basically a short sequence that is used to uniquely label a molecule. Okay, a molecule of nucleic acid. Okay, so this is different from the library indices and barcodes. Okay, if you remember, library indices and barcodes will tag all the molecules. I highlight all the molecules coming from the same sample with the same barcode. Okay, the UMI are basically labeling all the molecules coming from one sample with different sequences. Okay, so each molecule from one sample will have a different UMI identifier. Okay, and the UMI are usually found either in line, so next to the basically ligation boundary between adapters and your fragment, or next to the indices, okay? But again, I just wanna make sure that you understand the difference between UMI and the library indices and barcodes. Library indices and barcodes are tagging all the molecules from one library with the same sequence. UMI will tag each molecule with a different sequence that is coming from the same library, okay? So if we apply the UMI to the previous example, we actually have two different type of UMI design. The simple one is just insert a random sequence, so it can be like 8 random nucleotides. So in this case, you can have UMI1, UMI2, UMI3, UMI4, UMI5, and UMI6. Okay. So in this case, even if the fragments in red, yellow, and dark blue start and end at the same position, they actually have different UMIs, okay? So they're coming from different original molecules, okay? Which means that now I don't count 4 molecules anymore. I count 6 because I have 6 different UMI, okay? However, this approach has a problem, okay? So the random nucleotide design is not great because you can have sequencing errors, okay? So for example, if UMI2, the red one, and UMI3, the yellow one, they differ only from one nucleotide, the difference might not be biological. It's possible that these two was just like a sequencing error, okay? So that's the second design of the UMI. Instead of having random nucleotides, you can have pre-designed sequences, okay? So you basically design a ton of different sequences, okay? So you see that ATCGA is different from TAGGC, and it's different from TGCCG. And so since you know what type of UMI to expect, for example, if you look at UMI2, this is something that was not present in your original design, okay? So if it's not present in your original design, then you say, oh, it must be a sequencing error, okay? And then you look, what is the UMI that is most close to it? And it's exactly UMI3, okay? And it's just one nucleotide difference, okay? Usually when you do this pre-designed UMI, you have several differences between the sequences, okay? So now if we count again our original fragments, we have UMI1, and then UMI2 and UMI3 are exactly the same, even if it's a sequencing error. And then UMI4, 5, and 6. So the true total number of molecules is actually 5, okay? So this is just a way of saying, you can do the PCR duplicate detection using in silico method, so no UMI information. However, and then you can have different UMI possibilities, random and pre-designed. However, it's possible that you will have different results just because you're using different type of information. I would say the best one, it's this one, the pre-designed UMI application. However, it's very expensive, okay? So I totally understand if people don't wanna switch to UMI pre-designed oligos, but this is actually the true way of understanding whether a sequence that starts and ends at the same position is a PCR duplicate or not, okay? And with this, we're gonna do an extra very short mini quiz, okay? I'm just gonna post, same as before, the quiz link in the chat. And with this, we are done. I'm gonna stay here for a few more minutes. Well, still half an hour to answer questions, still through the Google Forms, okay? I'll give you a few minutes to answer. I'm just gonna pause the recording and then I'm gonna come back here to answer your questions at about 420. Okay. Okay, so for whoever is done um, answering the mini quiz, again, it's really important that you do it for attendance. Otherwise, you will not be recognized. And also, I have an idea of whether I want to, talk about a little bit more in detail tomorrow if you didn't answer a question correctly. If you have questions, you can just type it now. Otherwise, I'll see you tomorrow. One thing, if you're typing in questions, just move um, your cell selection to a different cell. Otherwise, I will not be able to see it. All right. So I see a question from Michael. Best practices for dual index combinations and signal recovery from sequencer. What indices shouldn't be combined and why? I think, Michael, for this question, Illumina, both Illumina and NEB will have basically a table to tell you, this base should not be mixed with this base at the same, I'm talking about the same position. What I can tell you, what they usually do is they're telling you what type of adapters you can use depending on the kit the adapters are coming from. What I'm telling you right now is the best practice is to use unique dual indices. And the problems usually occurs when you have only a few libraries in the same lane. But if you have like many libraries, let's say more than 4 or more than 10 or more than 20 libraries in the same lane, then usually the question you were asking before, what indices shouldn't be combined, it doesn't apply. The reason why is that the Illumina machine wants to see as much complexity as possible at each base. So the ideal situation would be for each position of the sequencing cycle to see an equal amount of A, T, C, and G. Okay, so if you have biases because like all of your sequences are A or all of your sequences are T, then the software of the machine is starting to just wait, what's going on? So that's how the quality decreases during your sequencing run. Okay, so if you have the same base at the same position. I hope that answered your question. But again, we'll see tomorrow the best practice for indexing is going to be unique dual. Okay, and I'll show you tomorrow that will minimize the index misassignment. Okay. Question from Shuya. Sorry if I misspelled your name. Is it necessary for each DNA molecule to get a unique UMI? If so, how can we make sure? Okay. Yeah, no. I mean, yes and no. I mean, whenever you're like getting, usually the UMIs are introduced during adapter ligation. The most common design is the random design. Okay. Sorry. I forgot to share the screen so everybody can see the question. Sorry about that. Is it necessary for each DNA molecule to get a unique UMI? If so, how can we make sure? The answer is yes and no. As I was telling you, the most common design is the random. And so, you might have already the design with biases. So the random that is usually synthesized in the oligos is not true random. So you have biases there already. Like few sequences might be overrepresented. But whenever you are deduplicating using the UMI, you are basically using two information. You're using the UMI sequence itself, and you're also using the mapping position. So if you remember this, let's go back to the slide describing the UMI. you have the information here. You have the information on the positioning. So this is one information. And then you have the UMI information, okay? So the combined information of positioning and UMI will give you the uniqueness, okay? So let's say somewhere else, so for example, chromosome 2, 200,000, 203,000, you have UMI 6. Should you discard this molecule? No, because it's mapping into a different position. So the combination of mapping and UMI sequence are not the same. So it's still a unique molecule, okay? So it's impossible to basically assign a unique UMI to each DNA molecule unless you have, you know, so many UMIs that it's, you know, it's very unlikely that the same UMI will be used. But you know, it's basically impossible because you're doing these the addition of this UMI in solution, so it's not like a pre-patterned type of reaction. So it's basically impossible to do that. Okay, it's basically impossible to say, okay, this molecule gets this UMI, this molecule gets the other UMI, and but usually the combination of the positioning and the UMI sequence is unique enough to letting you distinguish the true PCR duplicates versus true biological duplicates. Okay. Hopefully I answered your question. Okay. Okay, I'll stay here until 4:30 if you have questions. Again, if there is a specific topic that you want to see tomorrow, you're welcome to just type that in. And then I'm going to try to include it in my slides. Otherwise, again, I'll see you tomorrow. Thank you for being here. Hopefully, I didn't annoy you too much. And then if you want to see improvements, for example, you know, Marco, I hate cahoots, you know, just get rid of them. Just please make a comment, okay? So we can work better together tomorrow, okay? Question 2.2, yes. All right, question 2.2. Oh yeah, that's right. That's right. Sorry about that. Yes. Continue from line 15. All right. Is there any possibility to affect the quality when I apply RNAs at the end? Usually, I treat the RNAs in the initial part of the prep, and it can be removed with repeating washing. However, I got the samples from others which look which looks RNA contaminated. Do I apply RNAs now? I've tried to find the answers from Google several times, but I failed. Yes. So if you wanna, if you think you have a contamination in your final libraries with RNA that you wanna get rid of, you can definitely you can do an RNase treatment. I usually suggest you to perform an additional purification using AMPure beads, for example, to just get rid completely of the RNase enzyme. They're usually very sturdy enzymes, they will stay in solution, active, even if you heat up your sample. I would do a AMPure beads purification or a column purification at the very after the RNase treatment to just get rid of the enzyme. Yeah, if you think that's a problem with your samples, you can definitely do that. No problem. All right. Okay, yeah, I have another question. All right, let's go back here. Question one. So each, are you referring to? Oh, sorry, the question of the quizzes. Sorry, yeah. Let me open the quiz. While there might be only one choice be answered wrong, since it affects the other sequences, the score would be all gone too. Yes. Usually manually corrected. So if you... Okay, so if you don't have any other questions, I will close the meeting, and I'll see you tomorrow. Awesome. All right.